Vivek, you can start.
All right, great. Well, welcome everyone to our fireside chat as part of World Salon series. Our conversation today is going to be focused on when the pipeline breaks, building ML infrastructure for biotech R&D. I'm very excited about this topic. It's one that I hope that alongside all of you, will learn quite a bit about from our panelists. So just as a reminder of kind of our format for today, we'll go through some quick introductions for myself and each of our panelists. I've got a set of starter questions to start the conversation, but we welcome obviously any questions in the chat from our audience as we go along. And then as we finish up the guided questions, we'll open up into a little bit more of an open and free-flowing conversation. So a little bit about myself. My name is Vivek Mittal. I'm a partner at Health Advances. We are a strategy consultancy focused exclusively on the healthcare industry. I specifically lead our biopharma practice, so work pretty closely with a range of biopharma companies on development and commercial strategy. And you can imagine that in today's day and age, the topic of AI, how to incorporate AI, are we AI ready, is a key question that we face on a regular basis. So excited to dive into that topic and really talk a little bit about where companies are at the forefront of incorporating AI into their discovery and development processes, and where we're seeing some big challenges, because obviously not every company is at the same level of scale or readiness along the way. So that's me. I'll pass it on to Jasmine. I think that's the first person I see on my screen to do her intro.
Sure. Yeah, hi everyone. My name is Jasmine, and I build AI infrastructure for precision medicine at Character Bioscience. My work focuses on making AI research reproducible across like genomics, medical imaging, and clinical data. And mostly we build around the phenotypic data that researchers can confidently, translate a complex biomedical data into reliable scientific discoveries. And my main agenda, or like why I'm excited about biosciences, share some of the infrastructure lessons that we have learned and how AI works with biosciences, a new area for me, and I'm pretty excited about it. So happy to hear and like learn more about you as well.
Great. Niels?
Yeah, hey everyone. Super excited to be here. I'm Niels Bantilan. I'm the chief ML engineer at Union.ai. We're a company that shepherds a open source project called Flight. Maybe familiar, maybe not familiar to folks, but it is fairly popular in the biotech and life sciences space. Flight is effectively a durable, infrastructure aware AI runtime. And part of that means orchestration. I have a pipeline, a bunch of steps. I need to make sure they run in the right compute requirements. There's a lot more there, but yeah, super excited to be here. We built Flight to be a general purpose, but turns out a lot of the architectural decisions and capabilities that the platform provides is very amenable to biotech set of problems around processing genome scale, data sets, images, multimodal. So happy to talk more about that. But I actually have a biology background. My major was in biology. I did developmental and immunology. So I was kind of the mouse guy at a prior lab in a previous life. So I know what it's like to do wet lab research, the trials and tribulations of not doing sterile technique and just having weeks of experiments go down the drain. So yeah.
I think we've all been there, unfortunately. Natan?
Sure. My name is Natan Silberman. I'm the CTO at Artera. The clinical problem that Artera solves is that when a patient is unfortunately diagnosed with cancer, which therapy is the right one for a particular patient is often a very ambiguous choice. Artera has a series of FDA-authorized tests that assess a patient's prognostic risk as well as therapeutic response, which therapy is the patient going to respond best to. and those are considered standard of care in the US today. My background is predominantly in the AI space. So I've been working in AI for about 20 years, have a PhD in NYU in the area, and spent stints at Google working on large-scale computer vision before for the last 8 years or so, applying myself to healthcare, initially in ultrasound and now in digital pathology. That's all about me.
Great. Joshua?
Sure. Josh is good. Hi, everybody. My name is Josh. I head up the compute team. That's software and data science here at Octane. I also run our sort of platform operations team. So Octane is a small molecule discovery company, and we're really focused on a modality right now called correctors. We think a lot about sort of novel small molecule MOAs, things that people didn't think were possible with small molecules traditionally. My background is not on the biology side, so I haven't spent a lot of time in the lab. My background is in computer science. I have a PhD from UC Berkeley, studying PopGen. And prior to joining Octane, I was at a diagnostics company, Invitae, heading the bioinformatics team for about 10 years.
And Sivan?
Yeah, I'll go, I guess, last. Sivan Bukovici, Chief Technology Officer and Chief Business Officer at Carriers. My background is computer science, similar to Josh and others here. I spent the last decade at Carriers leading the computational side of the family, as well as the partnerships. For those that are less familiar with what Carriers does, we are a liquid biopsy infectious disease diagnostic company. So we are partnering up with hospitals and providers to support the diagnosis of their patients, primarily focusing on immunocompromised patients. These people, folks that suffer from cancer, transplant patients, people with HIV, end up suffering disproportionately from hard to diagnose disease. In fact, building on Atan's story on cancer, a significant number of people suffering from cancer end up dying because of an infectious disease, and our technology really focuses on getting the best technology out there to diagnose and support the diagnosis of those patients.
Great. Okay. Well, thanks everyone for those introductions. I'm sure, given the varied experience of this team here, that it'll be a great conversation. So I appreciate the intros. I know the first question is posed to me, but I actually think I want to start with Jasmine just to get us going, and we can come back to me at maybe somewhere along the way here. And so, Jasmine, I know you talked a little bit about your work at Character. Obviously, you are integrating multiple different data sources, so genomics, imaging, et cetera. How do you think about the different data components and where there's an ability for kind of creep in the data or lack of reproducibility that might occur over time as the data sets expand?
I think that's a great question because not everyone understands how difficult the bioscience data could be. Because patients' data is kind of like collected for treatment of the patients and not really training the AI models. So when we work with data that's kind of like so different in like genomics, imaging data, and clinical data, it's kind of like the assumptions that we made when we connect them, right? So like all of those data points are collected on like a different timelines. Imaging scans could be done on like a different dates than how you visit the clinics and stuff. So all date of services or like how the patient has been treated during that time of period kind of like gives you a timeline, or like how you define the baselines for that treatment kind of like changes on like how you reproduce that data itself. So reproducibility in bioscience is completely different than how we see in different softwares and machine learning models, because here you are, you have its own challenges with the modality as well. When you have like a different devices involved, like how the scans are being captured, your computation changes as well. Your annotations around data also changes. Genomic data is like mostly structured, but the reference pipelines and like annotations around it can evolve within time. like how we get all the protein structures, how loci and SNPs are kind of like defined and stuff like that. It's completely different than how you understand one biomarker or one phenotype per se. And also, like clinical data is probably the noisiest, I would say, because it was like collected to treat patients and not train AI models again. So
Sure.
a biomarker result kind of like, depends not only on the data itself, but kind of like the cohort definitions, kind of like a temporal alignment that you have, or like the inclusion criteria for that cohort population, exclusion criteria, how the QC changes, what type of population you are interested in. Sometimes where data has been collected is also a primary, driven research could be just on like, if that population largely based off of the US population or something, then you will see lots of like demographic changes in the data, and what drug development has been like targeted audience or something could be very different. So like how machine learning models can evolve and learn from that genomics data, and how availability of those data is also kind of like the key pointers on like how you reproduce the same scientific discoveries within the same kind of like uh, prediction drug or something like that. And mostly kind of like one lesson like I have learned is kind of like the reproducibility isn't primarily a software problem. It's kind of like the how scientific modeling works. And also for me, the biggest shift was kind of like the infrastructure isn't just like data anymore. It's kind of like preserving the scientific meaning across like multiple modalities, right? On so like researchers can rely upon the biomarkers that being kind of like generated or like targeted. Like if scientifically, does it make sense to, hunt down or target some of the biomarkers? Like, does it make sense to look at some of the biomarkers that are not really directed to the eye disease or something, but it's still a focus of the study? So, like, how you design the scientific experiment is the more reproducibility question than the actual infrastructure around it. So it completely changes on like what problem we are kind of like focused on. And for us, it's mostly kind of like the combination of all things, and how the scientific nature of question becomes, and like how we build trust around, researchers or clinicians, even patients. Like some of the patients are like consented for the studies and stuff like that. So they also want to know if this medication works for them or not, like if they see a progression in the scans or not. So those are some of the things that I think that I closely work with, and I see the reproducibility in complete different light, I guess. So that's my probably 2 cents on that.
No, it's really interesting. And it sounds like in some respects you're comfortable with the reproducibility or maybe lack thereof as you understand the changing nature of the question that you're trying to ask.
Yeah, because like the needs changes, right? Sometimes you are targeting for, a clinical trial or something. Sometimes you are just want to understand the prediction of the drug, or like how the progression of the disease works. So the question changes, then the cohort population and the baseline workarounds will change as well. So your focus shift on what's important or the priority to understand the population, or like understand the disease itself. So sometimes there's like so many other research that can support and help you understand the disease progression, but that might not be relevant to the, raw data or something, because raw data comes in complete different format. They are not studying patient that has been included in the research itself. So like how one drug can work for like, Asian population might not work for other populations. So it could be, hit or miss kind of a target, I would say. So that's why reproducibility and modality are like, such a depth research areas as well in bioscience. And it's a key pointers on like what devices you work with, what kind of a data you work with as well.
Yeah, it's helpful.
Yeah.
Any comments from any other of the panelists on that topic? Before we move on.
I have a question. It looked like Niels might have a question. I'll start. Maybe mine is quick.
Sure.
What I heard from you, Jasmine, is that you're collecting or you're receiving really this multimodal data. You don't have a huge amount of control over how it's collected. And I imagine that there's a lot of context that either comes with that data or that gets layered on top of that data by scientists at Character. And I wonder how important preserving and passing through that context into the models is to the outcomes that you eventually and conclusions that you eventually arrive at. Because that seems like a really, like, especially when you don't get to control the experiment or the experiments that are producing the data that are going into your ML, that seems like just a really challenging problem that I think a lot of people are dealing with. And I'm just
Yeah.
curious how you think about that.
I mean, that's a real issue because we are working with real data, right? So when we expect longitudinal data for patient, it might not be there. Like if we expect some of the QC criteria to be like, 4 or 5 visits per year, but it might not happen. Like that patient might follow through or might not follow through that, QC criteria as well. So at that point, you really don't know, like from one batch to the next one, how much of the sample size is gonna change. So for that, the scientific calculation that we do in the machine learning is more important than whatever the sample size is. So it becomes more kind of like studying the patient in the long term and not really considering them for like, only for 6 months or something. So when you run the same pipelines or like same machine learning models after some time, you'll still see similar results that you expect. So it's really designed around how the scientific machine learning works rather than just like, changing on the go of the patient population, because the core question is studying the biomarkers, studying the genomics, studying how the progression of the disease happens. It's not really studying about the population per se or like the audience changing or per se. So that's how kind of like the control research experiment work. And even for the QC criteria or something, it can change as well. Like you can study smaller population and kind of like observe or follow through them for like multiple years as well. Like there are some clinics who can send patient for like 5 years study or something like that. So that gives you a room to kind of like study in a broader terms and have like more research questions around them to study in various slides as well.
Thank you.
Yep.
I had a quick question about reproducibility. I think there are many layers to that
Yeah.
concept and how to actually implement it. So there's, software layer, pinning your Python dependencies or R dependencies and containers and stuff. What are the differences and overlap you see when you think about scientific reproducibility? Much larger scope there, but are there overlaps and distinctions on how you think about and implement reproducibility at software and science layers?
Yeah, like first thing first is like data cleaning is such a, heavy task. So like any analysis can only work if the normalization has happened. So like from raw data to kind of like moving into the scientific data is completely different. Like how the PDF data comes in from clinics or something, then it gets into kind of like the extractor, goes into kind of like, the schema itself. And then after that, it has to be still get cleaned up for like various things. Like there's so many, like in the PDF, there could be a various type of data types. Like it could be an image, it could be a table, it could be like a doctor's note or something that's like handwritten, and that it needs to be translated into data that could be used for, any analysis or like just for like an observation or something. So that's like the first layer that has to go through. It doesn't matter what data analysis you are doing, but it still has to happen. So that's kind of the reproducibility still happens in a way. And after that, there's like a modality that happens where you stick your ground to kind of like, work through on like certain devices for the scans. where you know like, okay, this device have like a really good scans quality, that could give you a big cohort population. So like the quality thresholds for those image scans and stuff like that could give you a broader population to study. So those are some of the things that could be reproducible and you can control. And there's also kind of like some of the data analysis where we study about drug is where you understand the systemic drug, you understand the disease related drugs that are already out there in the market. You can build those cohort population and directly use them for different purposes. So like let's say there's like a complement inhibitor sub studies that has been done. There's like systemic drugs out there. There's like also like eye disease related like drugs that we are studying. So you already have those kind of data that could be used directly, just by directing your data analysis start from that cohort population. So those are some of the reproducibility that you can still do, and that can save a lot of time. And if that's in place, then you can easily, run your analysis from batch one to batch two or the whole year per se, and really study the patient progression from, start of the year to the 2 year or 3 year kind of like the QC criteria, and really see the difference in like, how this patient has been progressed from the moderate to severe diseases and stuff. So those are some of the things that we have been doing. There's also like lots of image processing models that we have like, trained them like and noted them as well. There's also a feedback loop that goes into it. So like every scan that's been passed through for like, eye scan or something, we have them trained on such a deeper level of annotations that you can study the diseases from the scan, and you can really see how the fovea and like how eye deteriorates from like, moderate to severe kind of cases. And you can really use those scans for like different diseases as well, just by the training the models correctly on the good quality data, I guess. And that all kind of like a big legwork, I would say, but it's still worth it.
Great.
Yeah.
So one of the things that I guess I take away from my reading on this, and even just some of the comments you've all made, is one of the early success or proof points is actually getting the data into, messy data into some kind of structured format that then your tools and models can ultimately be trained on, right? And I think we've seen a lot of companies spend time really building either their own proprietary data sets or acquiring data that they can then structure, which I think is really important.
Yeah.
I want to shift a little bit, maybe, and Josh, come to you on this idea of, so you're obviously building the tools, the model systems, and then handing those over to the biologists, chemists to ultimately use them to develop drug. Those folks always have very different skill sets. So I'm curious as to how that process works, what it looks like as they use those tools and push, the boundaries of what they're doing, and where there's challenges there, and that kind of dry to wet lab conversion, if you will.
Yeah, thanks. I think it's a really broad and interesting question, and maybe like I think it's maybe even like a little broader than you just put it, because I don't think we're always passing over tools anymore. I think the big change that's happened over the last year or so is that, every biologist and chemist now is a data scientist and a software engineer, and that means they're able to analyze their own data, they're able to iterate on their own hypotheses, they can build dashboards, they can build tools. And that's been totally, I just want to say, like, I think that's really exciting. It's been transformative here. Not just because things get done faster, but because when you have one person, that one scientist who can both think about generating the data and analyzing the data and having all of that context, I think that ultimately leads to better analyses and better decisions at the end. And I think like, running the compute team here, like the upshot for me is that suddenly like I've got a whole new team of data scientists, right? Now every scientist is a data scientist. And like, what does that mean? What does that mean for me and my team?
So you've expanded your pool of expertise.
That's right. And now I have to answer the question of like, well, what are the the bona fide hired software engineers and data scientists doing now? Um, and the way that I've been thinking about it is largely that we've been trying to the thing that I think is really important is to lift up the work of the scientists and to kind of like provide what they need and to really impart taste, right? I think the data scientists have a lot of training and have built up that taste in many ways to impart that taste at scale and also sort of to put guardrails into place so the analyses themselves and the subsequent decisions are reliable and correct, which it's very, I think we've all used these tools, the agentic tools enough to know that it's very easy to go in a completely wrong direction with things in a very confident way. Um, and just like maybe more mechanically, what I've observed is that if we don't do anything, if we're not prescriptive, if we don't provide paved paths, you sort of end up in this world where people are creating and sharing HTML documents and Python files over Slack and over email. They're exporting data sets from heaven knows where and including those alongside to load into those Python scripts. And you kind of like lose your provenance of everything. You have versions. It's kind of like what I imagine software development looked like before there was any concept of version control, where you're just sort of passing scripts around. And that's frequently, my experience as a bioinformatician, that's frequently what bioinformaticians will do if left to their own devices. They'll have a big directory of thousands of stubs that they come back to and pass around. So you can't always tell where a result came from, and authentication
traceability becomes a big challenge, I would imagine.
Traceability, correct. And I think that overall this is just really a threat to like the reliability of the analyses and the quality of the decisions that come out of them. So what maybe to come back to the question a little bit, we've been trying to close that gap with a few tactics. I don't think any of these are going to be surprising to this group. There's no, super high technology innovation here, but I think they're really practical things that we've found, ways to think about things that we've found very useful in practice. So there's three of them. The first one is, and Vivek, maybe to your point, right? We're not building as many, analyses on the compute team properly. We're not building as many analyses or visualization or front end applications that go to the scientists. We're expecting and desiring them to build those themselves. And so because of that, right, the interface has really become the data itself, right? So the data is the interface. And I by the data here, I really do mean like the underlying tables. Like we have made the choice to give scientists access to those underlying tables. And I think the critical thing that stems from that, and we've had some experiences that have led us down this path, is that because of that interface, that data, we want to treat that with just as much care as we would treat any API, so that an agent or a human can sort of grok it and effectively and correctly use that data. And so for us, I think that's meant thinking really hard about how to join and transform that data. That's like filtering, aggregation, and really above all, like it's simple, but it's still so true, like documenting what is in that table, what is each specific column. That's all documentation that should be layered on, and that makes the agentic systems work so much better, at least in our hands, to have that documentation. We use Databricks for a lot of this, and I might sound like a little bit of a Databricks shill here, but like we've found Databricks to be a super effective tool just to manage, kind of this system of ETLs that we've built up in order to build this data interface that we want the agents to use. So again, like I know that's nothing new, but I think what we've found is that, and these are all like best practices anyway, but what we've found is that the agentic workflows that we're trying to support have really accelerated our adoption of these practices. So that's the first thing. I think the second thing is maybe a little more on the tool side. So we definitely have tools, and like as always, we want to build tools and democratize the use of tools. I think the thing that's new, and we do that through the usual ways. We build APIs, we build command line interfaces for things. But the thing that we've really started to layer on recently is this skill, and I mean like an agentic skill here. And I think what that has done is that it's allowed us to standardize the way that those tools are used and sort of allow the experts who are authoring those skills. So think of like a computational chemist here who's authoring the skill to impart their taste. And so maybe like as an example, take something again, maybe on the computational chemistry side that's complicated, like molecular dynamics. That's a complicated simulation to run. There's lots of pitfalls. Even if you have an API for that, that doesn't give you all of the taste that a computational chemist would have. But once you add that skill, the computational chemist can encode when is it appropriate to use this? What are the limitations? What should you be looking for as you view the results for QC? How do you interpret the
There's still
results?
judgment involved, right? So
There's
imparting
still,
the
yeah,
judgment to
exactly.
this thing.
And you could even imagine a system where the skill says, if it looks like this, you should really reach out to like a bona fide computational chemist to get additional support. So but all of that could be in the skill. And I think what that has enabled us to do is like if it's the broader chemistry team who's using that, it allows the broader chemistry to use molecular dynamics to democratize that kind of workflow, but still in a way that sort of like preserves and scales at least some of the judgment and taste that the true experts have. And I think that's just been a really powerful thing that we've really in earnest adopted over the last few months, and I think it's helped a lot. And then final thing is, related to sort of the entropy situation. We really try to keep the entropy at bay with so many people developing independently. We try to at least give things a home. So for sort of ad hoc chemistry or bioinformatics analyses, that home tends to be Databricks. Databricks, one aspect, there's many aspects of Databricks, but one aspect of it is that it essentially functions as like a very fancy notebook server. I think lots of data scientists, I'm sure all of you really enjoy using notebooks. We've sort of adopted this notebook infrastructure, and doing it in Databricks is great because it sits right on top of the data that we talked about previously. And there's sort of a built-in AI assistant where you can build whatever analysis you want, or you can use Claude or GPT or whatever you want to do that work. But the key thing here is that it's a shared central location for all of this. So in many ways, it's like a collaborative environment that I think most people are used to, something like Google Docs, except now it's for scientific analyses where you can save and share. You get at least the beginnings of something like reproducibility and auditability and how various results were arrived at. I'm not saying that there's not more to do there, but I think there's at least the beginning of that. It's not like it's just a naked result anymore that gets sent over Slack from some script that no one is ever going to be able to run again. Right. And then there's another side of the home thing, which is like we also think a lot about building tools and applications. I don't think I need to tell anybody here about how kind of like incredibly good the language models are at generating HTML and JavaScript, but lots of our scientists want to build those kinds of tools. And so we built, and I think there's commercial products available for this too now, but we built a little app store. And essentially what that app store does is it just gives the creators of those tools a home for them. And it also gives them sort of access to the underlying data. It takes care of all of the authentication for the APIs and things like that. And so really what we want through this is for the creator of that application to really be thinking about the scientific business logic of what that application is doing. And then we want to take care of all of the plumbing, right? And it really is plumbing for them. And that's been a real hit with the scientists too. It's kind of fun, like to look at the app store and see all the things that different people have built and play around with it. So those are the three sort of practical things that we've done to kind of make progress in this direction. And I guess maybe I'll just wrap up by like reflecting again on how exciting it's been to watch the scientists dive in and get engaged with this stuff. A year ago, or maybe even like 6 months ago, there's a bunch of scientists who would have never even dreamed of opening an R notebook to run an analysis. They would have handed that off to the data science team and waited, a day or 2 days or 3 days to get back a result. And now they're conducting their own analyses in real time, and they're loving it. They're having so much fun. They're super empowered, and it's just really, I'm sure you guys all see the same thing, but it's just been really wonderful to see. And so we're really thinking again about how we can just continue to lift this up, but to do so in a way that is safe and reliable and continues to generate sort of mostly correct results.
But it sounds like at the core of it, though, is you're augmenting and ensuring a human in the loop across the right. It's not a situation where there isn't an opportunity for an expert to ultimately come in, adjudicate, or verify what's happening.
I think we're trying to build. I mean, when you say expert, I think it's hard, right? Because if a scientist wants to do a chemist wants to do a matched molecular pair analysis, but they're not well versed in that kind of cheminformatic method, they may not be functioning as an expert, and we're trying to give them the tools to do that anyway.
Right.
And we're trying to build a culture where where the scientists continue to learn, right? Where there's ongoing education, and I think
you're almost augmenting their capability in that
that's
scenario.
right. It's a little bit trust and verify, where it's like we want the scientists to continue to learn, and we want them to have the sense that like they're not an expert in this, and this is a useful thing to bring up in the chemistry meeting so that they can get other people's opinion and make sure that they're running these tools correctly. Right. But I anticipate as time goes on, people will learn, the skills and agents will continue to get better. And I do think that we can more fully, and this is the path that we're on, is like, how do we, we do want human in the loop now 100%, but like I think we're pushing, we're trying to get to higher and higher levels of abstraction where you can have less human interaction and trust that the right thing is happening under the surface. And that's going to be a long road, and it's going to be an uneven road, but like I think that's the direction we're trying to head.
Vanessa, you came off mute. Do you have some comments here?
Oh yeah, just a few quick thoughts. Josh, a lot of what you said really resonates with me, specifically when you talked about data as the interface. We see that, both, of course, in life sciences, but across other fields as well. And every time you remove a barrier between data and decision by LLMing skills away, we shorten the time to making some of those decisions. Having said that, I've seen AI, and I'm sure all of us have seen AI accelerate everyone. And by that, I mean those that did extremely well before AI and those that didn't do as well. And the difference between the two groups is that one of them is getting to the target faster, and the other group is still running in circles. When I look at matching problems, people, and AI, I really think about three criteria. Would the person know what are good questions? Would a person be able to tell if the answer is a step forward? And would they be able to say, okay, I think we're done with this direction? And whenever at least one of these questions is slightly off, you are getting a person with the impression of the ability to make progress and content that could be quite convincing and lengthy at times, but we're actually not making the progress that we're hoping to make. I would love to hear, and it sounds like you guys are investing in it, and the tool sounds amazing. Beyond building a culture that says, hey, please be aware, here are the limitations. Are there any additional tools you guys are implementing or considering to implement to say you should really stop here? Here are two people on your team that should have a look?
I don't think that we have. I don't think that we've gotten there in like a tools or automation way. I think it's largely through process and culture right now, where the expectation is that when an individual works towards something and does an analysis and comes to a conclusion, that then through the usual set of meetings floats towards a broader body who's going to be able to poke at it and ask questions. And I think to your point, right, like everyone has become very sensitized. Well, maybe I'll just speak for myself. I've become very sensitized to being handed documents that are as you said, long and convincing sounding, but are ultimately not reflecting an actual understanding of the topic that's being discussed, right? Like, and so I think that the expectation, again, this is a cultural thing and not which is maybe not what you're probing at, but I think there's an expectation that we're all collectively building here and elsewhere, that like you're accountable for the results that you generate and for understanding how those results were generated and why they're scientifically credible. And I think in many ways, then like the language models are a great enabler of that, but they don't guarantee that. And I don't have, to your question, I don't have a fantastic way, other than, more agents of sort of automated way of like checking that sort of thing.
So maybe as a quick follow-up on that, do you believe that organizations are experiencing a new flavor of denial of service attacks? And by that, I mean, I'm getting very long emails, and the documents that I'm reviewing are very long. And if you study where engineers are spending their time, they're writing less code and they're reviewing more of it. Are you observing, either within your company or your customers, a situation where a significant amount of time is now being spent on the review process and the refinement of those insights?
I think it's lumpy. And I think that as I said, like I think that our collective tolerance for that kind of thing is already starting to wane a little bit. I think there are people who use the tools effectively in order to augment what they can already do, and and those are typically not the people that are going to send you a long document that maybe even they themselves don't fully understand. But then I think there's another group of people that are less responsible users, let's say, that are going to do that kind of thing. And so I think that what we're I guess if you give me a soapbox, I'll stand on it, right? But like, I think that what we're kind of collectively learning right now is how to function in this like new environment, and what are the bounds of like acceptable behavior. And I think a lot of that is going to be a company culture thing, but I do think there are norms that are still being established in that regard. I don't know, Sivan, does that resonate with you? Does that sound right?
It does. I think that it's interesting to see how the community is coming together to figure out what to do with these incredible tools and true accelerators, both internally and through our products to our customers. But there's also all these areas that do benefit from significant attention, especially now as adoption is happening.
Yeah.
Joshua, you mentioned you're giving folks direct access to underlying tables. How do you handle data versioning?
Oh, good question. We have not thought deeply about data versioning yet. I guess we have in one sense, right? So we will often analyze and reanalyze data through kind of some of our core pipelines. But typically we think about that in a very simple way, which is that, sort of through those filtering and aggregation steps, we try to provide, or we will provide the version of the data, sort of like our gold tier data is going to be the currently accepted version. And we haven't yet gotten to a point where we are, maybe this is really a question about reproducibility. We haven't yet gotten to a point about where we're trying to reconstruct something that was from a prior version of the analysis. But my base expectation is that, yeah, I think we don't have, I certainly don't have like a super solid answer on that, other than what exists within the machine learning pipeline in terms of preserving its inputs. What is there Natan, I'm curious, is there something specific that you have in mind, or like how have you dealt with this?
Yeah, it's a good question. I think there's different approaches, and you might use a different approach depending on the sensitivity of the data. We also, as much as possible, try to give people a let's call it as direct access as possible. That being said, typically when people interact with data, they're not interacting quite with the underlying tables. They're always interacting with the snapshot, right? So at any given time, somebody will, hit the button and a snapshot is created, and that's a thing that they interact with. And the snapshot comes, with the timestamp and a whole bunch of other things. And then the snapshots themselves are access controlled. But it's very important for us for regulatory reasons, and then for publications, we want to reproduce, like, how did this number and this paper come about? We have to be able to trace back, like, you ran this particular analysis with this Git hash against this particular data snapshot version.
Yeah, that's really cool.
Yeah, I'm curious. I think, Nathan and Sivian, when you think about how your teams run, where you're trying to produce an FDA, approved or authorized test, how does that access, that use of the data, and what the research teams are doing differ relative to what Josh's team might be doing, where they're, it's more, I guess, it's regulated, but in a very different way?
Yeah, I mean,
it's a great question.
Just to be clear, like, we're using this for like preclinical stuff, right? So it is the regulatory structure is very different.
Right.
Yeah.
So how does that kind of change the dynamics of the data access, use cases, et cetera?
Yeah, I mean, I think, quite a bit, because of the regulatory landscape in which we operate, both in the US and internationally, right? Firstly, there's no agile development, right? We really have to think about, you build a product, then once it's built, you have to go, document it, submit it, and there may be months of waiting for a regulatory response. So really important that we sort of locked things down such that throughout the life cycle of this product, through regulatory submission and throughout its lifetime, it's reproducible. Secondly, when it comes to post-market surveillance, like actually identifying, actually understanding like how the tool is operating in practice is really important, that we're able to pinpoint, why why a model's making a particular prediction, to try to be able to reproduce it if there's any suspicion that something hasn't gone right. And the third is it's very important also for a combination of regulatory and compliance reasons that there's a very clean separation of concerns. So, for example, we don't want our AI scientists to be continuously, hammering our test sets, and to even, in many cases, have access to the evaluation results that we're going to go off and send to the FDA or to another regulatory body. So, long story short, it's really, really important for us because of the nature of the product that we're developing, that everything sort of start from a snapshot of data version. The data, the underlying data is sort of like a river, right? It's like constantly changing. But at any given time, somebody takes a snapshot of it.
They have a point-in-time version of that they've basically extracted.
Exactly right. And then any workflow that is applied to the data is itself reproducible. Again, thinking in mind, like both from the perspective of a regulatory body or publication, like we need to be able to trace back to how that number came about.
These are actually a set of, sorry.
Oh, yeah, I was just like just saying about like how the data has been kind of like changing. It's also kind of like it depends on who the audience is, right? Like sometimes not all the scientists have like the clear context on what type of data they could be looking at. There could be like a shared data, proprietary data that could go forward for like different study purposes. So we also have like a de-identified data set where there's like a certain limits or like the edge calculation caps and stuff like that, that kind of like gives them the clear context of the data, but not really information towards the patient itself. So there are like some of the guardrails that's already been there, and I think that's the practice probably should be there, and either you are being audited by FDA or not, but like that just like the good practice of like, who sees the data and in what form they are seeing it. And even like most reproducibility kind of like comes around kind of like nightly syncs of the data that happens. And also like the snapshot that you mentioned could also be generated on like ad hoc basis, right? Like it depends on where you work from or like, I know Europe has like some different data kind of like the regulations per se. So like how you share data is also kind of like changes. And most of the AI tools that's been used in US are not like widely spread into the world, and they are not like accessible to other population. So like you anyway you have to build a data set for that particular audience or something. The stakeholders are changing constantly. So yeah.
That's
Yeah,
right.
these are a set of problems that we've thought a lot about at Union when building out an orchestration platform, an AI runtime. Basically, you want to look at architectures that have the most granular, fine-tuned control over where a container, a piece of work runs. For full disclosure, Artera AI works with Union. We're love working with them because we can see meaningful things happening with the stuff we're building. But for example, if a pipeline has heterogeneous requirements, both for compute, but also for where it runs, like maybe you need data locality for where certain things need to be stored, and you need to process data in that way. But then maybe you abstract away the data enough so that you can do batch inference in a different region, Australia versus Europe or US, right? If you have a monolithic pipeline, it's just impossible. It's just a workload that you cannot do. Whereas if you have a tool, a pipelining tool where every single node can run in a different cluster, on a different cloud, GCP, Azure, or AWS, you can have these heterogeneous workloads. You can unlock that capability.
I would add something onto that. So indeed, we work with Union and we've had just a great experience. One of the requirements that we've seen, some in the US and especially in Europe, is indeed data locality. And it's really not a nice to have, it is a must to have, making sure that every single part of your workflow is ensuring that bytes do not leave particular data locality. But I would say one thing that we're seeing in addition to that is not just let's say regulatory requirements, but simply requirements that come from individual providers who, let's say, don't want data to leave particular regions or even their cluster.
Because of data agreement
So,
where you want to keep it, yeah.
Yeah, so I would say like having something, as Niels pointed out, that allows you to really control the individual atomic units of compute is just a real deal breaker.
Mm-hmm. I want to come back just to this idea of infrastructure, I guess, for a moment, because what you're describing is, as companies get to a certain level of pipeline or scale, their needs change, right? And I can imagine, Artera didn't come to you day one. There's at some point in your history and time that you decided, hey, we need external support. So maybe can you describe, where do you see, at what point do companies kind of bump up against what they've built internally, and when should they start thinking about going external?
Yeah, it's a great question. I mean, I think for us and maybe for many people, once you hit a certain level of scale, you start to see that you need to start hiring a whole nother team to support a bunch of the infrastructure that you need to support your product. And there's a real question around like, are we going to scale up a team? And it's really important for this team to be in-house. Like our ultimate product is going to be better because we in-house this team, or is this the kind of thing that whether we have this person whether we have a group in-house or whether a vendor is doing it, it makes no difference to our product. But I think ultimately those questions come up certainly. I think relatively early on, once you've hit a level of scale where you have to deal with not just sort of a research project, but something that is really encountering the reality of dealing with patients.
Yeah, we've seen this pattern play out across many of our customers and open source users, where effectively the infrastructure is in service of the science and the product, right? So there's a scale aspect to it. There's a product maturity, science maturity aspect to it. You don't want to over-engineer a solution when you don't even have a working solution that you've found based on your data, the models you're training. Performance doesn't meet your requirements. There's no point in investing in a heavier piece of infrastructure to support that. But once you've hit on something, once you've trained a model, you've come up with a AI or ML powered system, perhaps, or even just software system, you need to scale. You need to scale your compute, your people, the regional and regulatory requirements. That's when a tool like Flight really helps. I think we've seen a few forcing functions to this. The first I've already talked about is heterogeneity in both compute requirements and like the granularity of data access controls and data locality. People, I think most of you here will relate to if you have a small team and the understanding of the end-to-end pipeline fits in a brain, like it's not a big deal to have it in a single repo and organizing your software in that way. Notebooks are great. Even at scale, notebooks are great. I mean, there's a place for all of these tools at all levels of the product journey, I would say. But once you have a larger team of data scientists, ML people, like you need, as Josh said before, you need a place to like codify these things. And I think agents are a great forcing function as well. You're forced to write as token efficient of a thing that allows the agents to kind of look at the documentation pieces that you can programmatically run with MCPs and CLI tools. Another one is regulatory and really, and also like as Natan said, the customer requirements, the government requirements are another part of it. And then finally, lineage of artifacts. If you need to audit, okay, this prediction came from this model, which was trained on this data set, you really have to think about the data set version, because then you don't want to be kind of flopping around, like looking through S3, like scrolling through and like, oh my gosh, there's so many data sets in here. Like, how do I even reason about this? As well as cost. So a lot of cloud providers' way of measuring cost does not track with like the unit of compute that's like this semantic human readable thing of like training a model, right? There's egress costs, there's compute costs, there are a bunch of other costs that we have taken great pains to sort of try to combine and estimate together so that your model training and data processing units of compute have roughly pretty good estimates for how much they've cost, so you can like optimize things. But again, this all comes with product maturity. Once you have a good model and have a good system in place, how do you bring down costs? How do you comply? Those are the things when you're spending more time maintaining and building the infrastructure, and like the science is suffering, is roughly like kind of the qualitative, subjective threshold at which it's, I think, time to start looking at tools that service those pain points.
Yeah, that makes a lot of sense, right? So basically, when the ML is I guess preventing the science from proceeding, or the service of the ML is preventing the science from proceeding, you've probably gone past the point of needing external support.
Yeah, that's right.
Got it. Great. So I know we have a minute or two left here, and I still have questions for all of you. But Sivan, maybe I just ask you quickly? Again, Curious obviously has marketed product. You are thinking about adding new tests to the platform over time. How do you kind of ensure that you've got that same kind of fidelity and signal test by test by test, even though maybe the underlying sample may be the same?
Yeah, great question. And well, luckily, nature didn't change as fast as AI does. So there are definitely things that remain stable in our entire process. I'm going to build on something that Natan said earlier. We are not regulated by the FDA. It's a laboratory developed test. We have a CLIA-certified capitated lab, and our test is New York State Department of Health approved. That means that we cannot just push things to production. However, we can move much faster than we could have been moving through in other settings. So annually, we release about 10 different versions of our assay. Usually about half of those are analytically in nature. That means that in some way we are changing the test functionality or its performance, and to a lesser extent, elements related to its reliability or convenience. Within that world, where you do release at a higher cadence, either new functions. So, just the other week, we released a capability that allows us to bring antimicrobial resistance information 1 day sooner to the physician. This is huge. In infectious disease diagnostics, time really matters. You don't have the weeks or even the days to get the right answer. So how do we go about iterating so quickly? Similar to the other points made earlier, you do need to have a clear set of well-curated data that is used during the evaluation and the model development, and then a completely separate set that is being used for the validation of those models. And we keep those separated. We have separated teams that are allowed to look at these different types of data. The R&D organization really dives and tears the data apart in whichever way they want, but the actual validation of the outcome of that significant R&D process, that's being done separately. We have to document the plan ahead of time. We have a lab director that signs on it, then the execution of that plan, the summary of that report, and that's being signed before we push a new version to production. And post-production deployment, we have both the verification step, but also monitoring. And depending on the magnitude of the change that we are deploying, we scale the amount of monitoring that happens. So if this is a highly laser-focused release that addresses a smaller set of microbes and boosting the performance on those, then you mount a certain amount of monitoring. If it's a more substantial change, you get far more people involved and have a more significant process. We found that process to be an incredibly good fit for a fast-moving technology. I would hate to think that we're sitting on top of new ideas and new capabilities and people out there are not gaining access. And in fact, every time we look at a release, we talk about what would happen if we push it by 1 quarter. And we talk about the amount of people that will get negatively affected by the lack of access to these newer diagnoses. I know that if I have a family member sitting in a hospital, I just really want to make sure that the best possible minds and technology are serving that person. And that's our commitment. And I'm sure others here on the call have the same commitment. Within the laboratory developed test environment, we can move faster, and we have been doing so.
Right. Awesome, great. Well, so I know we're right up on our time, but I think maybe what I'm hearing from all of you in summary is data, and having access to data that you can structure and annotate is critical. Having systems in place that share appropriate levels based on function and need is important. Then obviously the infrastructure behind that to process are kind of a couple of the key elements here that all of you are working on to ensure you've got fidelity around. All right. Great. Well, thank you everyone for joining. Appreciate the time and the conversation, and have a great rest of the day.
Thank you.
Thanks so much.
everyone.
It was very
Thanks so
nice
much.
to meet you.
Yeah.
Bye-bye.
Nice to meet you all. Bye.